topic is clinical trial monitoring, and it's a very important topic. And I'm talking mostly about the responsibilities of the clinical trial monitors, or called CRAs, clinical research associates, and we'll be sharing what they exactly do, what the responsibilities are, and the tips and tricks, what I've learned over the years, and some like things you have to need to know in order to prepare as a site for the clinical trial monitoring visits. So, trial monitoring refers to the systematic process of overseeing and evaluating the conduct of a clinical trial to ensure that it is conducted in compliance with the protocol, regulatory requirements, and GCP. The primary goal of monitoring is to protect the rights, safety, and well-being of trial participants and to ensure reliability and integrity of the trial data. So this is the official definition. They want to let us know that the site is like conducting the study in compliance with the protocol and regulations and everything. But there are so many other things also happening in the industry, in the clinical study management, that is making sure that the study is conducted according to the protocol, according to regulatory requirements and GCP. So what is the unique perspective of the monitor? So monitor is basically hired by the sponsor who's paying money to the site to conduct the study so that they can collect the data and then submit the information to the agencies for approval. So they are basically primarily their loyalty is to the sponsor. Regulatory agencies like FDA, their responsibility is to the citizens of they make sure that the data they collect, everything, the patient safety dignity comes number one. Research ethics board loyalty is to the patients, so they make sure that the patients come first. Everything else is secondary. And so each agency is, although looking at the primary goal of protecting the rights, safety, and well-being of participants and to ensure reliability, integrity of data, but their loyalties lie a little bit differently. The loyalty of his clinical staff at the site is primarily towards the site. They make sure that things go on properly on the site, and then they collect data properly. Although everybody is trying to make sure that the information collected is clean, is reliable, and then nobody's harmed in the process. So that's the whole idea. So they also, monitors go on the site visits, and then they oversee the things happening on site. And it's not only these two things, they also look at if the site is doing the studies as per the the agreement with the sponsor. So they also look at other stuff, look at the exam rooms, how exam rooms are set up, is there enough equipment or not on site. So these are the things they look at to make sure that the sponsor is satisfied with the site. So they report to sponsors. So there are basically 8 different responsibilities, and they look at whenever they go to the site. One is the source data verification. Next is protocol compliance. Third is informed consent process. Then how the adverse events are being reported. Is the investigational product being handled properly or not? How is the data management happening on site? Is the site adhering to the regulations and guidelines? And they will create queries to ask the site for these, to resolve these questions which they have, and then there's a process of query resolution. So we'll go into details of each one of them. So source data verification is a critical component of trial monitoring that involves comparison of data recorded on CRFs with the original source documents. Now, there's a lot of confusion of the original source documents. Many people don't understand what is source document, and they confuse it. And it's even like experienced coordinators, researchers, or even the sponsors does not really fully understand what source document is. Source document is basically a document on which information is collected for the very first time about the medical information for the patient. So, like for example, if a patient comes to the clinic and then the doctor sees the patient in the clinic, what they do is they will enter all the information in the chart, in the electronic medical records, and that becomes a source document. they go and do the X-rays, and then the X-ray reports are fed into the electronic patient record system. That becomes a source document. Then they go and do the lab test, and then results are available. So that becomes a source document. And so when we copy that information into the forms or transmit otherwise, then they become the CRF, or depending on whatever it is needed for. But the information which is copied on from where was originally recorded is not a source document. So unless, for example, let's say patient comes for checking, getting the vitals and in the clinic. So you document that on a piece of paper, and that piece of paper on which it was first recorded is the source document. And you talk to the patient, and they give the concerns, discuss the concerns with you. So you document them on like it's audio recording. You have a audio recording of the patient concern. That is a source document because it was the first where it was originally documented. So this is important because what happened is there have been people who have been doing studies, and then they will make up fake patients. So there, as you go, I've seen a lot of conferences in ACRP, and there are some conferences like sessions on fraud conducted in clinical research. So one of the things that happened is a coordinator will make up a fake patient, and they say this patient came in, and falsify, create a false data, and then fed it into the system because they don't have any patients coming in. So the monitors will come and see, and they want to see the source document. And there's no original source document for these patients because he never came and saw the doctor. There's nothing in the records. So this process makes sure that you are collecting data from verified original patients, which are actually coming to clinic and getting treatment from the doctor. And so this is important from that perspective. That's why they come and see and check all these various medical records to make sure that the patient actually exists. So for that reason, they need like access to these documentation, and because of the privacy and confidentiality concerns, there are a lot of confusion on this aspect also. So the confusion is that they should not be granted access to the source document. So what happened is, like suppose a patient, like monitor cannot do their job if they are not given access to source documents. So there have been different practices, as you guys already working in the field know, that we redact the name, the MRN number when we send documents through email and stuff. But when they come on site, there is agreement which is made between the site and the monitor that they will keep all the data which they look at confidential. There's a confidential agreement they sign. So they are allowed to look at the name, age, date of birth, so that they can verify that with the the CRFs and with the and to make sure that the patient actually exists. And then that information is kept confidential. They cannot share it with anyone. So it is a false, people have been creating hoo-ha about like, oh, he came and saw the name of the patient, blah, blah, blah. But they are supposed to, because otherwise they can't verify. So these two things are very important. Like, what are source documents? And a monitor should be allowed to verify source documents. Number two, they can look at the name and personal information of the patients when they're conducting a monitoring visit. So then what is the purpose of the source data verification? To confirm the data on the CRF is accurate, and I just mentioned that. And it includes wide range of documented materials such as medical records, lab reports, imaging results, health records. The patient diary is also a source document, and anything else that's generated during the trial, for example, vitals documented. Then they systematically review the data on the CRFs and compares to the corresponding source documents. So to check the discrepancies, and they often find discrepancies. Why there's a discrepancy? The reason is because we are human, right? We are transcribing data from the EMR onto the CRF, and then from the CRF to the sponsor, electronic CRF. So normally the process goes is we collect the data from the medical records, and then we write it manually on the paper CRF. They're still not obsolete, and they're still existing. And then we copy that paper CRF onto the sponsored electronic CRF, where it is compiled in the sponsor servers, and then finally cleaned and locked and then analyzed later on. So because of this transcription process, then errors can creep in. So this is, they make sure that the information that is copied on is okay. For example, if you like 69 or 96, then things can get weird. So this diagram on the right is telling about the various things, like various components of risk-based monitoring. So in the next slide, I will show you what this means. So there are like 8 components: the initial cross-functional risk assessment, then there is ongoing cross-functional risk assessment, quality tolerance limits, key risk indicators, centralized monitoring, offsite, remote site monitoring, reduced source data review, and reduced source data verification. So earlier in previous times, like maybe 10 years from now, the monitor used to go on sites and check all the records of all the patients, and it was a very time-consuming process. It was very counterproductive. So what happened was, over the years, a new way of monitoring has been evolved, which is called risk-based monitoring. So they classify the sites into high risk versus low risk, and then they do more extensive like monitoring of the sites who are high risk, and then do minimal monitoring of sites which are low risk. They determine through a set of criteria we'll talk about as we go along. And also there's like a method called remote monitoring. So they don't have to go on the site always. They can review the documentation on wherever they are sitting at. For example, they are sitting in their office, and then they can log in. And then because of technology, I can share my screen, and then all the documents I have about patients, I can scan them into my system and then share my screen with the monitor, and then they can have a look and verify that everything is okay, everything is doing good. So the initial cross-functional risk assessment is that there are various departments who are working together, and they have a mitigation plan, because in clinical research project management, we have different types of risks, and then we create a plan to mitigate those risks. So they do initial risk assessment. Cross-functional means across different departments, across different groups of people who are working on clinical studies, and then they do the initial risk assessment. And this is one component of risk-based monitoring. The other is ongoing cross-functional risk assessment. This is a continuous risk assessment, more for high-risk sites than for low-risk sites. Then the quality tolerance limits, or the accepted quality standards, of like, we will not accept anything below this. So these are also fed into the system by the monitoring plan created, and they have these QTLs. Then we have key risk indicators, like the metrics used to assess the performance. For example, a site is having high enrollment or low enrollment. Low enrollment is a risk indicator, or there are a lot of protocol deviations happening on site. So, or somebody died, on site while on the treatment, and or there is high turnover of the staff, and then there's no consistent, ongoing. So these are risk indicators. They will indicate that this is a high risk or low risk site. Then the centralized monitoring is like a central location where all the data from the sites is fed in, and then it is analyzed, and then they will evaluate what exactly needs to be done, make a plan. This is offsite remote site monitoring is same. It's a component of the remote monitoring, and then it's a link to the centralized monitoring. So then there is reduced source data review. So source data review is looking at the original source document to see they're authentic. Source data verification is like comparison of the data from the source documents to the CRFs. So these are all components of the risk-based monitoring, and I will show you a trend which is happening recently. So monitors look at all this stuff, and then they focus on the informed consent documentation in source data verification. They look at the eligibility criteria. They will do informed consent documentation of 100%, every single patient who is irrespective. They're not going to pick and choose. They will come and look, every single patient has signed consent, and they look at the consent process. But the patient was given enough timing, they have any discussions, all the different elements of consent was explained to the patients or not. So you have to have a consent in place, as well as you have to have a consent process in place, and how it was done, and the date and time it was signed, all that stuff. They check at eligibility and exclusion criteria, if patients who are in the study are actually should be in study or not. They look at the medical history, the existing conditions, look at study interventions, and how the treatment was given. They look at any adverse events happening, especially the serious adverse events. They look at very thoroughly, because that's the key safety component of the studies. Look at the lab results and vital signs, and the patient reported outcome. For example, there's a quality of life study, and then they are collecting information from the patients. So they will look at all these PROs and the questionnaires. If the patient actually documented these, then any other assessments and endpoints. So they can be conducted through on-site visits, as I mentioned earlier, physically examining the source documents, or through remote monitoring. So here is a graph. It was from a journal article I was looking at, and they would, is Association of Clinical Research Professionals. So they were looking at the data, and then they were looking at the trends, like on all the 8 components I just mentioned previously, what's happening in those in remote monitoring. So it's like quite a concept has been for some time, but in ICH GCP2, the new edition which came in, I think it was in 2020 or something. So they mandated to have a risk-based monitoring plan incorporated in the protocol itself, and then they encouraged risk-based monitoring. So these are the studies on the top, are studies which are ongoing, started prior to 2019, and these are on the one below are the studies which were started after 2019. So they were initiated in 2019, 20, 21. So this is the 3 bars are 19, 20, 21. And so they say here in the top one, it's very small, but I can tell you that initial risk-based assessment was around 50%, and ongoing was also around 50%, which has gone up to 83% to 89% in the ongoing years. So the initial and ongoing risk assessment has increased. At least one component they incorporate to make sure that the risk is assessed. Then the acceptance standards have increased. You can't see the figures here, but the numbers have increased. And then the you can see there's an increase in the key risk indicators, KRIs. So they have incorporating them to make sure that the sites are having high risk sites are defined early on, and then they go and do the assessments. Then the centralized monitoring has increased as remote monitoring has become more popular, and because it helps them spend less money, and then it's only go to places where it's required. So the centralized monitoring and remote monitoring, these numbers have increased for these. Then we have they also reduce the amount of source data review, so they will pick only select amount of charts to look at, and also the same goes with the SDV. So SDR is data review, and SDV is verification. So they will not go through all the documents on all sites. So they will go to because of the key risk indicators, they will look at certain elements of the in the site that indicate that this site high risk. So they will select a few patients. For example, let's say there was a patient, his initial lab values were abnormal, and then he missed 2 visits, and then the coordinator document that lab values abnormal, 2 visits are missed, and then they made an unscheduled visit to collect the data. So that becomes a high risk patient, and then they will come and they will look at every single thing about that patient. So it will be all the SDR, all these data review, and all the verification of every single document that's there. And if same goes to patient, like suppose he was admitted in the hospital, and then the monitor will come and look at all the documentation, everything about that patient. So, and then if that is in place, everything about that patient, these two patients is in place, is good. This means his side is a low risk side because they are following the protocol, following the compliance, and then nothing to worry about, that they're doing the job well, and then they will be labeled as green, like the low risk side. If there are problems, if they're not documenting things properly, then what's going to happen is they will um, like label this as a yellow or the red or high risk side. The STV findings and queries, if the monitor identifies discrepancies or inconsistencies during the STV process, there is queries with the study site to seek clarification or corrections. These queries are documented and tracked until they are resolved. So, so they will be raised, as I mentioned, raise queries and ask the site to explain themselves what is happening. And I also mentioned this, there is a shift towards risk-based monitoring. So this is targeted risk-focused approach. Now, I saw a movie some time ago, and it's a Oscar winning movie. They had like the embassy, was under attack by the local population, and then they shredded all the documents, before they left, and they escaped the embassy because they were going to be killed. And the locals came, and then they, strip by strip, they joined, the shredded pages and to create a document back. And that they identified who the people are who were there, and then so that they can go and kill them. So this reminds me, the monitor is like that. If you have a shredded document, he or she is gonna join them piece by piece to see what exactly was there. They're so tough, and like and then they will go and look at and find out what wrong was going on in the site. So it reminds me of that. Then we have protocol compliance in so we discovered the SDV. The next responsibility of monitor is to look at the protocol compliance. So protocol compliance in clinical trial monitoring refers to the extent to which the investigators and study staff adhere to the study protocol, which is a detailed plan outlining how the trial will be conducted. Now, understanding the protocol serves as the roadmap for the entire clinical trial. So it includes detailed information about trial objectives, study design, eligibility. So basically tells you how the study is to be conducted. And so, when you don't follow the protocol based on, and then what do we call it? It's called protocol deviation. So protocol deviation, how to manage a protocol deviation, how do monitors or the sites manage it. So this is another article, journal article, and in which you prepare for, for the, their inputs happening and outputs happening, and then you identify and see what are the discrepancies. Then you confirm and you review and analyze discrepancies, and then you close out. So I'll explain in detail as we go along. So it involves studies conducted exactly as described. This includes enrolling eligible participants, administering treatments correctly, collecting accurate and complete data, and following all specific procedures. So what is the importance of compliance? Why do we need to follow the protocol, verbatim, like word by word the same? It's important because if we don't do that, then the data quality is compromised. You're not gonna get, you're gonna get errors. For example, if let's say protocol says you enroll 100 patients, if you enroll 50 patients, then the power of the calculations is not good enough to justify that the treatment is effective or not. Or you don't follow the eligibility criteria, and then you have patients in them which are random patients, and then they don't have all those set criteria. So this means the data is not clean and cannot be applied to that particular population set, because there were other people also in there. So, and then so it affects the participant safety down the line, because if you incorporate that treatment down further down the line, and then these people will have a lot of problems because or may have side effects of that treatment. So the process is the event a protocol deviation? If yes or no, if it's and how you classify it, and it is major or minor. So there are two types of deviation, like which is significant, affecting patient safety, and the other ones which are not affecting patient safety. Okay, so the next one, common protocol compliance issues. So what kind of protocol deviations are happening on sites mostly? So the number one is the eligible participants. So people enroll participants and then they don't look at all the eligibility criteria. They're like intentionally or unintentionally missed. So for example, the patient is coming in and he's taking a drug or a treatment which was not not really it was forbidden in the protocol. For example, he's on a steroid and he's not supposed to be on any concomitant medications containing steroids. And then he didn't tell you while he was on, when he was getting enrolled. And then you later found out in second visit 2 or 3. So that becomes a protocol deviation because patient is on something which is not supposed to be, and you didn't know about it because he didn't tell you. And then so this is the most common. And sometimes people overlook, and they sometimes have a huge list of criteria, like 15, 20, 30 criteria to fulfill, and you have to check every single one of them to see if it's Then we have treatment administration, like if the medication is not given on time, if medication is not given as per the required dosage, then the assessments. So, for example, follow visits are to be done in a specific time period, and so assessment. So if you don't conduct the assessments on time, specific times, and then it's a deviation. So that we missed a patient visit. Then collecting accurate data, complete data. Temporal means like it is collected at the time when the patient visits was there, not before or after. Then do follow-ups and timely reporting of adverse events. So adverse events reported on time, like as they happen. So there's a time limit of 24 hours to 48 hours for the SAE to be reported to the sponsor. And if you miss a timeline, then it's a deviation. So all these things are commonly happening on sites. Then the monitors will look at the protocol compliance. So when the monitor will come on site, they'll look at all these things. So the deviation can happen on the protocol level, like minor issues. Then it can happen at the intermediate level, like treatment, therapeutics, or it can happen at the organization level. Like there is a SOP, the people are not following the SOP, or there is no standard operating procedure in place to conduct the way things the way it should be done. And then you need to create SOP. So they will come back, they will review the deviation, they will come back, and then they will tell you what exactly is required. Like so what corrective and preventative action CAPA plan should be. So whenever deviation happens, the site has to prepare a CAPA, corrective and preventative action plan, and they will um, ensure that it doesn't happen again in future. Sometimes the deviations are bound to happen. Sometimes, like I've seen in cases in studies that you go and deviation is coming, and then you report the sponsor ahead of time. They say, this is what happened. And for example, a patient is going out of the country for 3 months, and then he won't be able to come for next 2 visits, and is unavoidable. He has to go. So you report that to the sponsor ahead of time, that this patient is going this and it's going to be a deviation, and these are the reasons. And then so you are secure in your position. You're secure in your situation that you told them already this is going to happen. So nobody's going to create any problems for you. So risk-based approach, as I mentioned earlier, focuses on the areas with highest impact on trial outcomes. So there are like a lot of issues on the right. You see there are a lot of issues, and then you filter down those issues to the ones that matter, and then you fix those issues. The related to patient safety, integrity, they get prioritized in dealing with it. Compliance with edit review, trust in clinical research processes. So protocol compliance is fundamental to the success of clinical trial, and that is ensured by the monitors. Then the next one is the informed consent process. So the informed consent process is a critical aspect of trial that involves obtaining voluntary and informed agreement from participants. So this is a big challenge, like recruiting patients in studies. Now we have like a coordinator like this. Now who would say no, joining the study? So the thing is, that's why probably the PIs hire young, like sexy looking coordinators, they can lure patients into the studies and stuff. But like the jokes apart, like the influence of what the coordinator has on the patients determines how many patients they go on the study. So they will have to trust the the system. They have to trust the person they're talking to, and it like the person who's enrolling them has to show respect for the people and respect the autonomic individuals to make decision that they should participate in research or not. Now there are the key elements of informed consent. Every like a coordinator who give informed consent to the patient should make sure that the purpose of the study is explained, the procedures, what will the patient will undergo when they are on the study, like what the schedule of visits will be, what are the risk benefits involved in participating in the study, should be mentioned. What are the alternate treatments if they don't want to participate in this study for this treatment? What are the options available for them? Then all the data will be confidential, stored, and who will look at the data, like the authorized people from FDA, Health Canada, the immediate care team can look at those data. Then the participation is voluntary, and they can withdraw anytime. The contact information of the coordinators, the investigators, and also study staff, the ethics board should be mentioned. If they have any issues, they can contact them. And final, like any financial aspect, if they're getting any compensation, any parking reimbursements, or any daily meals for the time being spent on the study. So the process of informed consent is initial discussion. Then we, provide the document to the patient, and then have them to look at the document and review. And then we respond to all the questions they have, clarification. It can be the questions asked by the patient or the immediate loved ones, like, somebody who's accompanying the patient will also ask these questions. Then we make sure that they understand, like we ask some questions to see they are okay to come on these scheduled visits, they're okay to take this medication, they know what the side effects can be, and they're okay to go ahead with that. And then we ask them to sign it, date it, time it, and then you sign and give them a copy for future reference, and then keep one original in your binder for the monitor to come and see. So monitor will review if all of this was done on site or not. And it's important because there have been studies conducted when patients have they have without any consent, they were asked to go through torturous treatments, and it was during Nazi war and all that stuff. So there's a huge history of maltreatment. Even in modern times, like, some psychedelic drugs were given, hallucinogens were given to prisoners, and then they had like traumatic experiences down the line, and no consent was taken from them. So you can't create a human being as a guinea pig. You have to give them autonomy to make sure that they can say no anytime they want to. So then the another one is the special considerations in the vulnerable population. So children, pregnant women, prisoners, they're autonomy is compromised because they are dependent on someone who is the authority figure and in charge of taking care of them. And if they are scared that if they say no, that this can affect their well-being. So this is important. Um, and people with cognitive impairments cannot make decisions on their own. So they're having, their information like shared with other people. Like, information like, for example, their like decision-making capacity is in hands of somebody else who's take care of them. So there are some tools to make sure that the vulnerable population is given enough information. So the low income one, set clear expectations. The patients who are not educated, you make sure that they understand. Employ an interpreter to deliver the consent in their own language if they cannot read the document. Uh, and then the subordinates, like if I have seen like the in a pharmacy institute, the principal investigator was the lead pharmacist, and he was conducting the drug studies on students. So that is not right thing to do. So when you know recruitment not performed directly by supervisor, so somebody who is not directly your superior should ask you to conduct a study on you. So that is important. Then childbearing and potential childbearing women should have minimal and preventable risks. And also pregnant women should have low risks on fetal health. This is a challenging area because unless you do studies, you don't know if it's gonna be creating any risk or not. Same goes with studies in kids. So in people, um, like you do not know if drug is safe or not, and then you extrapolate the doses to the body weight, for in children. So there is a big debate going on. Should more studies be conducted in kids or not, and how to get the consent process for those children? And the parents agree to it, and should we ask the kid if they agree to it? And what if the kid says no and parents say yes? And what if the kids say yes and the parents says no? So it's a very complicated matter. These are the debates happening in the ethical realm. So the research ethics people are the ones who are. So you always can always talk to an expert in the field and what is ethically correct for me to do. Should I go forward with this? Should I enroll this or not? And they will give you feedback on that. Then you have AIDS, HIV. So their confidentiality has to be maintained rigorously because this can lead to job loss and loss of insurance benefits and stuff like that. Then gender diverse persons, balancing needs of corresponding sex and gender identity. Now there is a equality being created, but now the US government is creating new laws, which are against the LGBTQIA population. So now their representation into the clinical trials is also very limited. And another thing is, I sometimes think, genetically they're either male or female, or so. It's very hard to, like, for example, if somebody who is had done operation to change the gender, from male to female. So should that person be included under males in study conducted, or should that person be included study conducted females? Because they have hormones in them, but the genetic component is XY, and now they have modified them to look like XX. So it's ethical dilemma, which is very difficult to answer. Should that patient be included because his biologic systems, kidney, livers, etc., might excrete as per, like as a male person is handling the drug. So this is complicated stuff. And then obesity and chronic diseases. So in those patients, no hopes should be raised that they're going to lose weight by participating in trial. And then in migrants, you have to give an interpreter facility available so that they can talk to them in their own language. And impaired mental status. So evaluate cognitive competence, and then make sure that the person in charge wants the best for that patient. Next, the thing the monitors look at is the adverse event reporting. So it's a crucial aspect of clinical studies that involves documenting and managing any untoward side effects. And they can range from mild discomfort to serious medical complications. So by monitoring this, make sure that the patients are safe. So there are different types of AEs, like serious and non-serious side effects. So serious more demand more attention, and there's a process to collect this, and there is a process to document. I have done a separate presentation on how to report SAEs and how to report adverse events in different populations. And there's a process to collect SAE and transmit it from the site to the sponsor, and then there's a way the sponsor collects the SAE from all the sites, and then he transmits it to the regulatory agencies. And there's a way by the regulatory agencies collect all the information from all the different sponsors, and then they relay that to public. So that's a whole different topic in itself. There are investigator responsibilities and sponsor responsibilities. So investigators are responsible for assessing and documenting AEs, and their task is to report them as early as possible in as much detail as possible. And they also assign the causality, that whether this is due to the medication or not. And sponsor will review, analyze, and then report to the regulatory agencies as required. So the reporting process is initial assessment, and then there is documentation of the SAE, then causality, whether it is related or unrelated, anticipated, unanticipated, serious, non-serious, and then report to the authorities. And SAE has to be reported to ethics board as well. So they will also look at the data, and then the DSMB boards will look at all the SAEs and make sure that study needs to be continued or not. And then you follow them up. So once you collect all information, send it, then you follow them until they're resolved. So if the SAE is not resolved, you go all the way down to the very end of the study. And then, so it's important that a close timeout, like a closing point is documented, that this SAE started or AE started on this date. This was what was done, treatment was given, and then it is conducted, and then it's stopped on this day. Like after this, the AE was resolved and no longer followed. Okay, so pharmacovigilance department deals with this. So they will come and look at all the safety data, like all the data collected. Sponsor will analyze it, and then decisions are made. So there's a pharmacovigilance department who is, collecting AEs, SAEs about a particular event. So there's an officer, pharmacovigilance officer, designated in the company, who is getting information from everywhere, and he also do literature searches. He also reviews all the information that is out there in the media and public, and then he creates safety reports based on this. So they're writing safety reports, writing their getting information. They're advisors on what to do. Usually medical personnel or with medical background. These are called medical monitors. Medical monitors usually have an MD, and they are the way expert in the ways of clinical studies, clinical research, and they are on sponsor side, and they look at all the information. So pharmacovigilance expert and medical monitors are slightly different. Medical monitors advise on the like all these overview or look of how these studies are conducted and if they're facing any side, if patients are facing side effects. Pharmacovigilance officer collect all the information and then they write reports and stuff. They do not probably advise on this stuff. The next area in which the monitors look at is the investigational product handling. So they make sure that the drug is properly stored, received, dispensed, administered, and is accounted for. So, you have to maintain logs like when the IP is received. So upon investigational product is checked for accuracy, condition, and compliance, then IP is stored under appropriate conditions. Storage conditions are monitored regularly to prevent degradation or contamination. There has to be temperature controlled environment in which the drug is supposed to be stored. Then we have to document how much medication was given to each participant. Track the quantity, how much the patient received back, when was it administered, and all that. So there are various logs you have to maintain, and they will go through all these logs. They will even come and count the number of pills remaining and before they are sent off for destruction. So they will come and look at the logs. They say, okay, this much was given to patient, this much they returned back, and they will sit down and they will count, how many pills are there. So this is important. So because there was another session attended in ACRP in the fraud, and the coordinator was selling the medication off, for profit. So he was getting this trial medication. So he, and he was cited. So he was blacklisted. He cannot participate in trial anymore anywhere in US. So he used to receive medication and then he used to, not give it to the patients or falsify that given to patients and then used to sell the medication off. So there are things happening, that's why all these measures and checks came into place because people have been doing all sort of things outside. So reconciliation happens, then there is destruction. So there's a destruction certificate that you get from the pharmacy who destroyed your medication. The number is mentioned, name is mentioned, date is mentioned. You keep it in the record to make sure that so monitors will come and check that. They say, okay, either you return this to us or destroy it. So that will be determined early on in the clinical protocol. Like the medication will be returned back unused or it will be destroyed on site. So you establish that even before the study starts. And when it's established, you talk to the pharmacy, get check, like how much is the cost gonna be, for destruction. So you negotiate the cost with them, include that in the budget, and then you, when the medication is is due, then you destroy. So Aisha, if you have any questions, you can stop me and ask me, as I go along, or you can type it in the chat.
No, I'm good so far.
Okay, awesome.
It's somewhat familiar to me, so I'm
Great.
just following along, and it's a good refresher for me. Thank you for.
Good. Thanks. So you guys are not sleeping. That's great. Okay, awesome. Then there is an unblinding, like emergency unblinding happens. So suppose in double-blind studies, the investigator doesn't know what is happening, whether the patient got the actual treatment or not. So if an SAE happens, then there are certain codes, that we can break the blind and then see what the patient is getting so that they can determine causality to it or not. If there is a causality, if the patient was actually getting the experimental drug and then he is going to see side effect, then it has to be notified to all the sites that this is, for example, somebody got a stroke while on the medication and you break the blind, then you inform all the sites and the study is halted unless you collect more data, more information, so that no more patients, will actually get stroke down the line because of this. Then, as I mentioned earlier, temperature monitoring. The products are sensitive to temperature fluctuations. And then you have to, like, for example, the device shown here can look at, it's like all remote monitored. So there are like automatic logging of the temperature, and then you just go into the logs every day and look at any deviation. There are alarms set up. They will inform the person in charge, what's happening. But this is all in compliance with the regulation ISO GCP. So all the staff need to be trained, will be handling the investigational product or medical devices. And quality control bases are there, a good record keeping, organized records, is good. So quality control is for the like the end product, like on the is product focused. I always used to get confused between control and assurance. I always used to say which is which, like, and assurance is the prevention. Assurance is the set of SOPs and procedures in place so that the quality is maintained. But when you test the final product and you find a defect in that, and then you go and change the QA processes, quality assurance, because the end result is not good. So this is ongoing process. So they make QA procedures in place, and then they test the product. The product is okay, then QA is okay. If end product is showing problems, and you go back to QA. And every batch of drug which is manufactured in the industry is tested. Some of it is tested for the standards. That was in CMC, chemistry manufacturing control of regulatory affairs. So you get the product, which is each batch is tested. So make sure that the QA processes are in place. Everything is according to the standards. Then the data management, data collection. These methods are specified in the study protocol, and so there's a data management plan which is put in place. Now there used to be old paradigm was ECRF. So people used to collect information and put it on site, like put it on a server or something. That used to just have the data. That used to double entry, put in paper form. Some sites are still doing that. But the other new method is the all data is put in a clinical trial platform. Now the technology is advanced and they have made platform integrated, like electronic trial master files, ECRF, everything all integrated with each other. So this has a better control. There's a separate presentation which I gave on these ones. So they will come and look at the CRFs, paper CRFs, ECRFs, find discrepancies, see how the data is collected, is good or not. And then they will validate the data, do range checks, logic checks, consistency checks, and they will mention, if like this is more like a job of data managers, not the monitors, but they will find discrepancies and they will ask the sites of why the discrepancies are. And these discrepancies can be if the data is not up to date, if there is any duplication, if there is any wrongly labeled ID, like one patient data is in another patient chart, any missing values, any numerical outliers. If the units are not consistent, sometimes it's milligrams, sometimes it's gram, somewhere it's kilograms, and sometimes it is pounds per region. So they will look at all this stuff and make sure that it's consistent, reliable. So once the data is, submitted, all studies complete, then it will be cleaned, and then it will be cleaned by means missing data is furnished, and then it is standardized and normalized. So as I mentioned, that all the units, everything is okay, and then they will give on values of either 0, 1, 2, 3, 4, or 5. So to make sure that statisticians can do the analysis on that data, so it's normalized, and then it is submitted for review and approval. Also, when you collect all the data after the data is analyzed and submitted, it is sent to archiving. So it is stored for like 25 years in Canada, because the Health Canada can come and review the data anytime, and then is there any discrepancies? Because sometimes the long-term effects of medications are not known. It can appear 10 years, 15 years after you took the medication. Data management. So data management practices must adhere to regulatory guidelines such as GCP, ensure data integrity, accuracy, and personal protection. Then technology, they are like different software and tools required which they use, and there are certain EDC systems. So they are aware of what you're using on site, and they also have expertise on it. So, for example, site is using Epic for clinical, like electronic patient records, Epic. So they will be expert in that. They can review it. They can know how it is. So good data is accurate, is complete, is consistent, is relevant, is entered contemporaneously at the same time when it was collected, is valid, and is uniform throughout. These are the ALCOA guidelines for the data quality. So data is a management is a meticulous and systemic process that ensures the accuracy, reliability, and integrity. It involves various steps from data collection and validation to analysis and archiving, all of which contribute to producing valid and credible trial results. If the data is not cleaned, then it cannot be verified for analysis. The analysis is biased, and all the money and time is spent on conducting the study on site is a waste. So every component of clinical trial go hand in hand with each other. One cannot exist without other. So if there's no recruitment, there is no patient coming in. If there is no patient coming in, there's no data. If the data is not clean, not clear. So all these steps have to be consistent. So monitors make sure that all these things are consistent, happening properly on site, and then that the study is conducted as is desired. So another thing they look at is are the studies conducted according to regulatory guidelines as for the Health Canada guidelines, and US FDA regulations, or whichever jurisdiction the study is happening under. So multinational studies are happening in various countries, like in USA, Canada. So in US, it will be according to US FDA regulation. In Canada, it will be to Health Canada regulations, and accordingly. So the monitors going to each site will make sure that study is conducted according to those regulations, according to those jurisdictions. And also, it goes without saying that the ethics board are chipping into their, and they will make sure that ethical guidelines are followed, and the GCP is being followed. The same thing. So everybody has to be trained on that. Everybody has to be trained on the ethics guidelines. So the query resolution, so they have come, they looked at all those things which I just mentioned, and then they will create queries, say ethical queries, which will be released to the site, and then the site will respond, and the response is documented and then reviewed. And if the response is satisfactory, then the query is closed. And this is a process which is repeated endlessly until all the data is clean. That's the key thing, key tool which they have to make sure that the site is compliant. So they will create a query, get response, document the response, approve the response, and then file it. And then again, get another query. So it's very annoying for the client. I've gone through it. So it's annoying for them to, like, if the site has so many queries. So sometimes you just answer and then you say, no, not acceptable. And then there's certain software or programs which doesn't make it easy because there's no two-way communication. So two-way free communication is good. So I used to pick up my phone and call, hey, so you send this query and blah, blah, blah. And then I used to discuss, because it's very limited on the software to mention. So you always, that's idea is to pick up the phone, call them. I got these queries, and I what do you want? But is this okay? Should I put this in the system? Will that be all right? So that's what you used to do, and then they were satisfied. The talking always helps to solve problems, instead of just putting information in the system. So they identified, various stages of data review, including data entry, validation, monitoring. So it can arise because of incomplete or illegible entries, inconsistent data, outliers, missing information, or deviations from study protocol. Query generation and documentation. So monitors, data managers, or research associates raise queries based on identified discrepancies. As I mentioned earlier, so they will create, this is the process like so on the right. So the submission is from patients, lab vendors, you submit the data, investigators, and partner CRO, they collect the data. Then a part, operational control, make sure it's as per the standards, and then the clinical programming and data management gets the data is clean for safety, and then these biostatisticians, they collect the data and check safety monitoring, and this analysis is done. So this is the cycle. So this is the process. So create, send, track, resolve. If not resolve, requery, create a new one, and then finally when resolve, update the events. But also some queries are more sensitive and critical, so they need to be responded. The best way a site can maintain a good relationship with the sponsor is to answer the queries as promptly as possible. If you get a query and you answer it right away within a day or so, then your relationship with the sponsor and monitor is good. If you take weeks, or like don't respond, they keep emailing you or calling you and you don't really care about it, then your relationship is going to go back with the. So I used to tell people like the best way to maintain good relationship with sponsor is to good communication. Let them know what's happening, why. If we are not able to get to it this week, you say I'm very busy this week. I'll get back to it next week. I will check the talk to the patient, get the response you need. I'll get back to you. So I'll report to you this. So they are satisfied. They know he got my email, he acknowledged it. He said, okay, he's gonna work on it, and then they can wait. But if you get the email, you don't respond it for the whole week, and you're doing the same thing as previously, but you don't tell them, and they get upset. I sent you an email last week, you didn't reply back to me. So what's happening around this? So communication is very important. Then follow-ups are required. If the thing is a happy monitor is a happy sponsor. That's how it goes. Because the sponsor is delegating all the responsibilities to the monitors to represent them on the site. And if you make sure that they're happy, then the sponsor is most likely very happy, and there's not going to be a problem. So it's the same thing, in clinical research, everything is a repetition because you look from the site perspective, you see things from the monitor perspective, and it's looking at the same thing over and over again. So people get fed up, when they see all these things coming over and over again. And all these things are interlinked to each other as well. So you mention one thing in one component, and then you mention the same thing in other component. For example, I mentioned ICF in patient recruitment, I mentioned ICF in monitoring, I mentioned ICF in eligibility, I mentioned ICF in query resolution. So the same thing, So that was just presentation on monitoring.